So we're now in our last session of day 2, and we're going to now be continuing with that theme of practical solutions. And for this session, we will have 2 speakers' presentations, and then that will be followed by the question and answer session. And our first speaker of this session is Parinaz Mahbod. Please share your screen, Parinaz.
Can you see?
Yes, I can.
Okay.
You already have it up. Thank you. Parinaz is a senior research assistant in Dr. James Herman's laboratory at the University of Cincinnati College of Medicine, Department of Pharmacology and Systems Physiology. She earned a master's degree in genetics from Science and Research University, Iran. In 2012, she joined Drs. Tong and D'Alessio's lab in the Division of Endocrinology, Diabetes, and Metabolism at the University of Cincinnati. The focus of her research in their lab was the role of ghrelin on glucose metabolism. In 2015, she joined Dr. Herman's lab, where her research emphasis shifted to studying central nervous system stress circuits and the impact of stress on experimental reproducibility in rodents, and the role of specific molecular species in stress regulation and pathology by studying the novel genes and microRNAs involved in stress integration, using gene knockdown and gain-of-function methods with conditional knockout and transgenic mice and viral vectors. Her presentation today is on how mouse handling limits the impact of stress on metabolic endpoints. Welcome, Parinaz.
Thank you so much, and good evening, everyone. Today, I'm going to talk about series of neuroendocrine data to indicate that minimizing stress by modified handling method in male black six mice not only will improve the animal's welfare, it will also help researchers to obtain more reliable and reproducible data by stabilizing.
Parinaz, I'm sorry
Yes.
to interrupt you. We're having a little trouble listening to you, so if you can speak just a little louder, we'll be better.
Sure. It will also help researchers to obtain more reliable and reproducible data by stabilizing animals' behavior and physiological responses to the environmental stress. Why did we start this project at the first place? Two routine experiments that most endocrine labs perform to study the effect of different elements in glucose metabolism are GTP or glucose tolerances and ITT or insulin tolerances. Basically, they give animals glucose or insulin and monitor their glucose levels in particular time points. These 2 tests are very sensitive and critical indexes to monitor animals' response to some manipulation in certain peptide and receptor genes, or the effect of administering a specific kind of drug. And more importantly, animals' reaction after some surgeries, like gastric bypass surgeries, or even combination of all those factors. Therefore, having a reproducible data in GTP and ITP are extremely important in many endocrine labs, but it is also challenging to maintain the high put. Why? Because glucose level can fluctuate in response to stress. In fact, there are prior studies note that stress stimulates glucose release. To reach the goal of having a perfect GTP and ITP test, we decided to look for ways to reduce methodological flaws and reduce variability in the experiments. Initially, Dr. Eric Smith in our lab invested in methodological flaws, and he found a handful of evidences about the effect of handling animals in outcome of experiments. For example, Hurst and West paper that was published in Nature Method on 2020 compared a standard method of tail picking mice with using of tunnels or open hand. They found out that tail pick method will induce high anxiety, while using tunnels or open hand led voluntarily approach and lower anxiety level. My presentation today is focusing on the importance of voluntarily approach. We decided to develop small experiments to study impact of Hearst and this suggested method in output of routine tests in the lab, copying mice instead of tail picking them. Parallel with this study, we also looked at the effect of spending more time to interact with mice in the outcome of our experiment as well, by holding them in our hand and giving a gentle backbone massage to habituate them to the personnel and basically minimizing variable environmental stress that could be caused by human health. To differentiate this method from Hearst and West cupping method, we called it massage cup method. This method was slightly different than Hearst and West method, only by increasing the interaction time. The training procedure lasts for 2 weeks, minimum of 5 days a week and once or twice a day. Each session is between 2 to 5 minutes interaction. Usually the first day is the most challenging one, and we spend more time to comfort animal. If it's the first time that you want to try this method, using a cylinder to cup the mice will be extremely helpful to habituate. But by the end of the third day, only 2 minutes of interaction would be enough. In the next slide, I am going to show you the training procedure and the reaction of mice and the second day of training. I realized that the position we hold the mouse in our hand plays an important role to speed up training procedure. That means holding them in a consistent position, let them habituate very quickly. As you see in this video, I move the mouse in my hand 2 or 3 times, grab it in that particular position that I'm talking about, then start to give a gentle massage. You should have a control over your mice, but at the same time, your mice must feel it is still free and secure. As you will see, while I'm covering the mice by my right hand, my fingers are loose. And when it pokes any spot in my hand with its nose, I let mouse get out from the exact same spot and explore around. It's amazing. It's like making some kind of communication. After this maneuver, they usually come back to my hand and even consider it as a safe place to hide and they not even try to escape. As I said earlier, we made this video on the second day of training, but as you see, this mouse is already habituated to me and we can almost consider it as a trained mouse. So it's not a really hard procedure to perform. Now you may ask, when do we know that our mice is trained? The answer is you will notice it very soon. But anecdotally, there are some signs that help us to judge if we have done a great job in training our mice. The first one is urination and defecation rate. If you have worked with mice, you know that as soon as you grab them, they start urinating. Well, trained mice don't urinate that much, when you grab or hold them. They have shinier and healthier fur. They sit on your hand and basically they are very cooperative during your experiment. Even when you inject them, they don't exhibit aggressive-like behavior as long as you handle them only by their designated method. Mice have different reactions to different people, like most of the animals. But if you teach your personnel how to handle your mice by using cup massage method, or even only just cup them instead of tail picking them, you will notice that your mice will respond equally to all your trained personnel, and it will eventually reduce the variability in your data. I was also asked, do we really need to train the mice for 2 weeks? Well, when we started this studies, we wanted to have a solid protocol to follow, and we get a great data using it. So never tried to change it fundamentally. However, based on my experience and observation, I think usually only 5 days of consistent training would be enough. I would rather to have 2 training sessions the first 2 days, but we can reduce it only to 1 session per day after second day. If you are willing to take this approach, I strongly recommend to continue to handle between 1 or 2 times a week by the end of your study. So the point that I am trying to make here is continue your interaction with your mice, especially if someone else is in charge of changing the cages. Make sure you also have some interaction with your mice. You can even ask your wet take to handle them by just cuffing them instead of tail kicking them. I worked with room techs that followed this instruction for some of my studies, and it was very helpful. In this slide, I just want to briefly introduce the behavior test that we use to study anxiety-like behavior in mice. The elevated plus maze made of a plus-shaped maze elevated above the floor with 2 open arms and 2 closed arms. Animals which exhibit less anxiety-like behavior spend more time in the open arm and vice versa. The light dark box composed of light and dark chambers. Animals spend less time in bright chamber, has higher anxiety-like behavior in comparison to mice which spend more time in the light chamber. Open wheel test is an apparatus to assay general locomotor activity, anxiety, and willingness to explore in animals. Mice restrainer is another non-invasive device that we use to induce anxiety-like behavior and assess the change of corticosteroid level in blood in response to acute stress. But what is corticosteroid? Corticosteroid is a steroid hormone produced in cortex of adrenal glands, and plasma corticosteroid is considered the main glucocorticoid involved in regulation of the stress response in rodents. It has pretty similar role to cortisol in humans. Next slide, I'm going to show some data that we obtained from testing the effect of Hurst-West method on both behavior and metabolic endpoints. In this study, we trained 2 groups of our mice with cuffing them versus tail kicking them for 2 weeks, and then expose them to EPM and assess their anxiety-like behavior and blood glucose and cortisol level. As it is indicated in this graph, cuff mice spend significantly more time in open arm than tail kicking mice. And also, they have a higher total numbers of entry into the open arm. We didn't see any differences in total distance travel in maze between 2 handling methods, which indicate that it's a good thing and to indicate that there is no sudden hyperactivity in either groups. This data shows that tail-kicked mice has higher level of anxiety than cuffed mice. This set of behavior data also confirms Hurst and West method worked on our hands because we reproduce similar anxiolytic effect of cuffing mice. Earlier, we talked about increasing reproducibility of data. Recently, I found a paper was published just 2 months ago, and they represent EPM data that was interesting to me, and I thought it may worth to share with you. According to UNO, if I pronounce the name right, repeatedly handled mice following Hurst and West method spend more time in open arm, and they had more open arm entries. Means they found reduced anxiety-like behavior in their handled mice. It's comparable sets of data to the result that I just presented in my previous slide, confirming how this method produced similar data in different labs with different personnel. Hurst and West didn't show the effect of their method in metabolic endpoint, which was important for our lab. So while we exposed our mice to 5 minutes EPM, We collected blood immediately after EPM and 30 minutes after EPM to study the effect of Hurst and West cupping handling method in both glucose and cord level after EPM stress. This data shows how cupping method reduces stress-induced glucose level following EPM exposure in 30 minutes. But there was no significant difference between handling of two groups on plasma HbA responses to EPM. After revealing that Hurst and West method reduces stress-induced glucose level in cupped mice, for further investing the effect of this model and this method on metabolic endpoints, we handled two groups of diet-induced obese mice by cupping them versus healthy team for 2 weeks and performed IPGTT day 14 after fasting them for 6 minutes. We found no differences in baseline glucose level between these two groups of mice after overnight fasting. However, after administering IP glucose, glycemic response in tail-tipped mice was significantly higher than the cup group that was reflecting lower glucose tolerance in group of mice that were handled by the standard tail-tipped method. As I described in detail earlier, we also made a slight modification in Hurst and West method and called it cup massage. And we performed sets of behavioral experiments to assess the effect of this modification in behavior and metabolic endpoints. For this reason, we trained our mice with cup massage method, and we had a control group that were tail-tipped for 2 weeks, and then exposed them to open field and EPM tests and assess their cord level after EPM. As you see, cup massage animal spend more time in center of EPM. Latency was less than healthy animals, and again, locomotor activity, there was no difference between locomotor activity. And more importantly, these cup massage animals spend a longer time in open arm than healthy animals, and also open arm entry has difference too. This data shows cup massage handling, in other words, increased interaction time with mice, significantly reduced anxiety-like behavior in male mice. The cup massage method not only has anxiolytic effect, it also improves metabolic outcomes. As it's demonstrated in this graph, the cup massage method reduces blood glucose in fasted mice compared with tail-tipped mice. Also, we found that corticosterone level was reduced in both 5 minutes and 30 minutes following EPM in a cup massage group. This data implies that anxiolytic effect of interacting with mice in both behavior and physiological level. So after having all this nice data, we decided to continue practicing this handling method in our future GTP to limit glucose fluctuation due to stress and reduce the variability to have a consistent data. As you see here, in this glucose data, we have very small level bar in cup massage groups and even in cord data. So it was remarkable data that we found. However, when I was trying to handle one of our knockout mice by cup massage method, I observed something new and very interesting. I noticed this knockout mice don't habituate handle. I was almost able to predict their genotype only based on their behavior. So we asked for consultation from stress group specialists that were living by Dr. James Gammon to find an answer to this mystery, and we started collaborating with this group. But first question was, what is this knockout line? It was Ghrelin knockout, and Ghrelin gene was mutated in this line. So the next question is, what is Ghrelin, and what does Ghrelin do? Ghrelin, or hunger hormone, is a 28 amino acid polypeptide that mainly secretes by stomach, also released in smaller amount by small intestine, pancreas, and brain. Ghrelin has many functions. It's well known to stimulate appetite, increase food intake, and promote fat storage. But after this observation, I also find that there are multiple publications about the effect of this hormone in stress, anxiety, and depression. But not all of them agree with each other about this function of Ghrelin. Also, sometimes they are totally different. Well, plasma Ghrelin circulates in both of the acylated and acylated forms. The acylation occurs through Ghrelin or acyl transferase or GOT enzyme, and the only known receptor for Ghrelin is growth hormone secretagogue receptor or GHSR. But the biology of this hormone is complex. In this presentation, I'm not going through detailed info about what is known about this hormone and how Ghrelin works, because as we see in this nice diagram, Ghrelin has a complex mechanism of going through every step and discuss about the pathways that it is involved. Will require longer presentation. However, we have more detailed discussion in our paper to refer. But here is, in this specific study, our focus was evaluating the impact of Ghrelin on stress and anxiety that were revealed by our modifying handling method, and we were looking for an answer to the question that why we couldn't train Ghrelin knockout mice even after 2 weeks of handling. Does Ghrelin have an anxiolytic effect? To answer this question, we bred 2 cohorts of Ghrelin knockout mice and their determined wild type controls, and handled one cohort with using Pathobiology method, and the other cohort was healthy. Then we exposed them to a series of behavior tests. The behavior data from this set of our experiment was very interesting and confirmed that the observation we had previously. We found that both non-handled wild type and Ghrelin knockout mice spent almost the same amount of time in open arm of EPM and light chamber of dark blood flow. Means there was no difference in anxiety-like behavior between non-handled wild type and Ghrelin knockout animal. But on the other hand, we found significant difference between handled wild type and Ghrelin knockout mice in the amount of time they spend in open arm of the EPM and light chamber of the light dark blood. So what does it mean? It means handling has an anxiolytic effect on wild type mice, but we couldn't detect the same phenotype in Ghrelin knockout handled mice even after handling because of lack of Ghrelin. This finding also is compatible with our previous observation. As I said, we used to have Ghrelin knockout mice in our lab for a while, but never noticed this behavior until we start to training them and found that, okay, there is difference in behavior between wild type and Ghrelin knockout mice. It means Ghrelin had an anxiolytic effect that was masked in non-handled animals, but handling mice revealed this phenotype by reducing the anxiety level. We didn't see this. We didn't see any differences in locomotor activity in neither of the cohorts. But another interesting piece of our finding is end exploration. That is compatible with the open arm times in panel A, but what does it mean and why it is fascinating? Recording this behavior data, I noticed some of my mice explored the entire open arm, which is a rare behavior in mice. Analyzing data later revealed that all of these mice belong to wild-type handle group. It was another interesting observation because it's unusual for wild-type mice to walk along the entire open arm. They usually only walk halfway through the open arm, then go back to the center, go back to the closed arm, and then try again. That was fascinating to me to see how handled wild-type mice willing to explore the entire open arm. But it was also interesting that handled Ghrelin KO mice didn't exhibit this risk-taking behavior and were acting like non-handled wild-type. This slide demonstrates HPA responses to acute stress that was induced by restraining our mice for 13 minutes. Panel A here shows that our Ghrelin KO mice have a higher core in 30 minutes after stress in comparison to their wild-type control, which is another confirmation that Ghrelin has anxiolytic effect, even if we couldn't detect its behavior in non-handled cohort. However, we don't see the same effect in handled cohort. Perhaps handling reduced the effect of stress in both genotypes. So as a result, the core level was not elevated enough to demonstrate these differences. Comparing this data with behavior data implies that non-handled cohorts have a high anxiety level that didn't let either of wild-type or Ghrelin KO mice explore open arm or light bugs. But as it is indicated in core data, in handled cohort of animals, due to lower level of anxiety, due to level of stress, wild-type mice start exploring the open arm and light bugs. But since Ghrelin also has anxiolytic effect, we need to have an intact Ghrelin system to reduce anxiety in a level that might start explore in open arm. That's why handle Ghrelin KO mice still spend less time in open arm. In other words, both handling and Ghrelin are required to see the behavior phenotype. Looking closely at baseline core data was also interesting. It shows handling reduced variability in baseline core level in wild-type mice, and we see a very small error bar and less scattered data. This slide I'm presenting the behavior data that we collected to confirm the anxiolytic effect of Ghrelin in male mice by treating them with both acylated and acylated form of the Ghrelin. To see if giving the hormone back to knockout mice will retrieve the anxiolytic effect of Ghrelin. We trained 3 cohorts of Ghrelin knockout and their beta-mate wild-type controls for 3 weeks, following 1 week of mud injection. We didn't really inject them. We just habituated them to the injection position and put the injection site. At the end of 3rd week, we treated our cohorts either with saline, beta-ghrelin, and EXOGENOUS. Our finding implies 3 important points here. Even after injecting mice, we had a reproducible data. Ghrelin KO mice that only received saline exhibited a higher anxiety-like behavior compared to the wild-type control by spending less time in the open arm. That means handled mice that were exposed to injection are still collaborative and handling effect is maintained even after injection. In other words, handling can reduce the stress after injection and pain. Ghrelin knockout mice that receive either azoghrelin or dazoghreline spend more time in open arms in comparison to Ghrelin knockout mice that only receive saline. It confirms anxiolytic effect of Ghrelin peptide. We couldn't detect any of these effects without handling mice. So, in summary, treating experimental animals in handling reduce stress and consequently we feel less variability in distribution of our behavior and endocrine data. Less variability results in enhanced reproducibility of data, means reduction of number of the experimental animals that could be otherwise required to repeat their study. Mouse handling limits the impact of stress on metabolic endpoints, and mouse handling can unmask hidden behavioral and physiological characteristics. This work has been accomplished because of the great contribution of the stress and endocrinology lab. I want to use this opportunity to thank Dr. Herman, Chair of the Department of Pharmacology and System Physiology at the University of Cincinnati and the World Authority of Stress Neurobiology, for his generosity and contribution in this study, and Dr. Eric Smith, who is a remarkably knowledgeable mentor and walked me through many experiments. I also want to thank Dr. Tang for giving me the opportunity to be part of this, our research group, and everyone in our team who helped me to design, run, and publish this project. The financial support came from 3 different NIH grants. This cute picture was made with my daughter based on a joke in our lab about my handled mice. They say Parinaz wants blood from her mice. They voluntarily pull up their shirts, say, how much do you want this time? Thank you so much for coordinating the symposium and inviting me.
Thank you very much, Parinaz. And thank you also for sharing that lovely drawing from your daughter. It's really special.